Hello friends, welcome to lecture 3 of week 8. In the previous lecture we have looked at how to predict the genes which are the coding genes, protein coding genes. However, like in among all the coding regions that is basically there in different genomes, eukaryotic genomes particularly, a large proportion is the non-coding part. And some of those are very important to predict, such as tRNA, ribosomal RNA, microRNA, long non-coding RNA. So we will look at how these are predicted and what tools are used for these. So basically these are all the part of the transcriptome, that is the expressed region from the DNA, the region that transcribes, right? And some of that is expressed which are the mRNA which makes protein. And there is a large proportion which is non-coding. Okay, so basically this will help you to predict the expression level of different genes in case of coding, whereas also this will help to detect the microRNAs and other regulatory RNAs which are responsible for regulating various pathways, genes which are there in the cell, right? So basically, like there are approaches like MicroRNA, RNA-seq which are used for this, and the methodology to process the total transcriptome may vary from the objective. Like if you are going to just sequence the mRNA, right, the functional part, okay, the protein coding part, or the regulatory part which is the non-coding other non-coding RNA, so that may vary. So like typically, as I said, there could be like miRNA, siRNA, right? There could be mRNAs, alternate splice mRNAs, and other type of RNAs in the cell, and all these are the components of the total transcriptome that we extract from any cell. For example, in this case, this is a pluripotent cell, right? So non-coding RNA are not translated into protein, and but they do a lot of important regulatory functions, right? And in addition to regulatory, there is a structural RNA component which is basically the tRNA and ribosomal RNA, transfer RNA and ribosomal RNA, okay, which are very important and that you might have read so far in detail, right? So we will be seeing that how we can predict these, okay. So these are called as housekeeping non-coding RNAs, the tRNA, ribosomal RNA, because they are required in the cell at all the time for the protein synthesis to happen. And the other part is regulatory non-coding RNA, right, which is in which we will look at the microRNA today, okay. So among the non-coding ribosomal RNA, the large proportion which is housekeeping non-coding regulatory RNA include tRNA and ribosomal RNA, okay. Which forms these are structural RNAs and are important for the basically the protein synthesis, right? And in the regulatory non-coding RNAs, we will look at the microRNA, although there is piRNA, siRNA, and non-coding RNA also, which have their own functions, right? So like this picture can tell, basically out of the total transcriptome, if you see, right, a large proportion is the non-coding RNA. That means when you extract the transcriptome, only 2 to 3% will be actual coding RNA, that is the messenger RNA, and rest other will be the non-coding RNAs, including the, of which the major proportion is still the structural RNA, right, that is tRNA and ribosomal RNA. Okay, so coming to tRNA, basically its role is very important because it brings the respective amino acid to the protein synthesis site, okay, and it basically facilitates the physical link between the mRNA and the amino acid, okay. So and it is a structural RNA. You can see that it is forming this clover leaf structure, okay, with stem and loops, right? And there is an N2 codon loop which actually binds to the amino acid and brings it there, and this loop also follows the wobble. So basically for 61 amino acids you expect 61 transfer RNA, because one transfer RNA per amino acid, but there could be less than 61 tRNA in genomes, okay, which is a common observation. and because of this wobbling, because one can act at bring multiple, and also the number of genes that code for it are not like 61 tRNA 61 genes, there could be much more than that. Okay, so basically what it does is there is this anticodon leaf which actually brings the amino acid to the peptide synthesis site. Okay, this is the ribosome which is doing the peptide synthesis. So basically these two, you can see that this is codon, and the third codon is basically it is bringing here, right. So this is how it brings the amino acids and extend the peptide chain, okay, on the messenger RNA. So just to give you an idea of how many tRNA genomes may have or how it varies. So like C. elegans have about 620 genes for tRNA. Yeast has 275 genes, bacteria have 40 to 50 type of genes. So basically that means there are more number of genes per tRNA because it is much more than 61 that you expect, right. And all eukaryote for the mitochondria, basically 22 mitochondrial tRNA genes are there in most of the eukaryotes. Okay, human have more than 500 tRNA genes, okay, to code for the different 61 codons. And as I mentioned, like you expect like 61 genes, at least 61 different type of genes for 61 codons, but it is not the case, it is much lesser. Okay, for example 31 tRNAs are required to translate unambiguously all the 61 so minimum 31 should be there, okay, and it could be like 40, 45, 50 and like that. Okay, so it may not be 61. So don't get confused if you see, note that basically all the number of tRNA, unique tRNAs are less than 61. Okay, so tRNA scan or tRNA scan SE Sean Eddy lab, okay, 2.0 the latest version. This is one of the oldest and still the most reliable and used tool for predicting the tRNA in the cell, right. So in the genome analysis also we download this software and analyze it locally. Okay, so basically it has a database also against which it's compared. So it has a module called Infernal now. Earlier it was Cove, now it is called Infernal. So basically it does two things, this software. It compares the potential tRNA with the target primary sequence, right, or the known primary sequences, right, and also evaluates the structure whether it is really a tRNA, because tRNA has a typical structure. Okay, and after that it gives a score to each tRNA and then it predicts the, like, this is the tRNA. And now in the new version it also can do the mitochondrial tRNA prediction. Okay, so this is the algorithm, but I have just explained that to you. Okay, so this is a web tool for tRNA scan SE, and you can upload your sequence or paste it here, right. But this is limited to 5 million base pair. That means you cannot do large genomes, but at least bacteria you can upload and you can predict the tRNA there. But it is simple installation and you can always do this locally also. So you will be selecting like what is the sequence source, basically eukaryote or bacteria or archaea, because it will be comparing, and the primary sequence is also predicting the structure. So it should know that which kind of organism you are dealing with, right. And then search mode could be default or this legacy that includes the tRNA scan and uFindRNA, and that is a Cove model, or the Infernal, which is recently it is using, and it is very slow, but basically it is more sensitive, right. And then you say run tRNA scan SE and this will give you the output. Okay. For the local installation, again, like as I mentioned, it is not difficult to install. So just say wget and this is the place from which you can find the tar-gz folder of tRNA scan, and the same way you will unzip and untar this tar-zxvf, the folder which is tar and gz. So basically Z is for gz, and basically tar is for untarring it. So basically it will unzip and untar it. So Z is for this, extract, verbose and file. Okay, so this is the thing. So then go to the directory okay, and then you do the this command /configure, and then it will configure the tool, okay, for your system, then say make and make install. So this will install this on your system, right? And but with this you also for 2.0 you need to download separately the infernal 1.1.2, which you can get from here, and follow the same instruction for its installation and download and installation, and you can use it. Okay, so use it locally. There are so many options which are available, which you can find using this tRNA scan minus options, okay, minus help. So we'll give you all the options, but if you the command that you may use is like tRNA scan. For example, you want to look for eukaryote, you can select -e for eukaryote. So here you can see -e for eukaryote, -v for bacteria, -a for archaea, -m for mitochondria. So you can specify anything here. Then -f, okay, which is here, okay, because you also want to check the structure of the tRNA. So you can do that also. -m, okay, which is here. So it will report all the stats for the tRNA that you are going to find, okay. Then your input FASTA file or multiple files. So it should have .fa extension, so it will automatically know that this is the input file, and -o for output. So you can specify any file here. Okay, so this is one of the output files. So it is giving you the input sequence name, okay, tRNA that it has found, right, their start and what it is, this is meant for the tRNA, the anticodon for that, and basically the other information, the score, right, which may be helpful, but it is already reporting you something which is above the threshold score. Okay, then this is this summary statistics. So basically it will give you all the information of what it does, and what are the where the file is stored, and right, and the different type of tRNA, the amino acids and their respective count. Like for example, in this case you can see that serine and arginine had the maximum count of the total tRNA genes it found, okay, and total it found was 745, and the split of that is given here, okay. And this is the linear secondary structure of one of the tRNA, and it's matched with the database. So in the different genomes, like after the prediction, this I'm providing you like some summary to just to know that how much was found. For example, in case of banyan tree, okay, Ficus bengalensis, the total tRNA was 679. In case of F. religiosa, that is people tree, it is 668, right. In case of turmeric, it found 335 miRNA. So basically it varies from genome to genome, right. And like compared to the coding genes, like if the it was 50,000 in this case and about 27,000 in these cases, right. Then ribosomal RNA is another structural RNA, which is actually very important for the protein synthesis, and this is the most abundant ribosomal RNA, and that is why you have to do the ribodepletion step when you prepare the library for the coding mRNA or coding RNA, because the majority will be the ribosomal RNA, okay. So basically it forms the various subunits which do the protein synthesis, the small and large subunits, right. And so to predict the ribosomal RNA, there are several tools. What the commonly available is or useful is BARNmap, which people are using these days. Another one could be rRNA, and there could be other tools also, but this is one of the more useful and comprehensive tool, okay. It is available in GitHub, and basically all these programs are available at GitHub, and most of the programs that are used by in computational genomics are available there, and you can download it from there, okay. So basically it can predict the ribosomal RNA in genomes, and including the bacterial 5s, 23s, 16s, archaea 5s, 5.8, 23 and 16, mitochondrial and eukaryotic. So basically it is including the prediction modules for all these different type of ribosomal RNA from different classes of genomes, okay. So it has now a NHMER which comes with HMMR for predicting the ribosomal RNA. Okay, so installation you will get it from GitHub. Okay, go to the directory and install it, and then basically you can check what the different options available by saying /barnap --help. Okay, so these are the different options, like you want to give complete chromosomal file, or defining the kingdom, like which bacteria eukaryote or something. So for example, in the command line which can be used, it could be barnap, the input file name, specify the kingdom --kingdom, okay, and then eukaryote --outseq out. So basically the kingdom name and the output folder name, and it will predict the ribosomal RNA for the input file and save it there. Okay, so these are the predicted ribosomal RNA sequences in FASTA format, which it gives as the output. Okay, so that means you are getting sequence as well as summary of the sequences where it is found in the genome, and which you can use it and also report it in your publication and use it further for the analysis. Then MicroRNA are one of the today most widely used or understood regulatory non-coding regulatory RNAs, which are small and generally like 21 23 nucleotides long, and they do regulate a lot of different types of mRNAs, and still their function is people are trying to know. So this is the typical process in which they are made, from the primary mRNA to pre-miRNA to the actually functional mRNA. Okay, and they bind to the various complementary mRNA. So it could not be a perfect match, but at least some good region should match to the mRNA for their successful binding and function. Okay, so basically there are so many tools to predict miRNA in genomes, but we will be looking at some of the standard tools that can be used here. Okay, so basically first understand what is microRNA. So these are like small non-coding RNAs, okay, which like generally regulate the expression of mRNA. Okay, and basically they should be like about 20 to 26 long reads. 2 long reads should be there. So you can see that this is one part and this is another part. Okay, 2 long reads are there. So you can see that there is a slight overhang also here, okay, by 1 or 2 base pairs. Okay, and this loop is also slightly not oriented aligned, it is a disaligned loop. Okay, and this is the other loop region, that is about 8 nucleotides. Generally it starts with AU, right. So basically in this I am just written the various properties of this. So basically it is 26 nucleotide long. Okay, they form 2-arm hairpin precursor, which is like this. Okay, right, and generally like the because there is a splicing by the or cutting by the various enzymes. So basically 5' end is conserved, and whereas the 3' end shows some changes or kind of overhang. Okay, so it at least 16 of the 22 nucleotide should show imperfect complementarity in the hairpin structure like this, right. Among these 20 at least 13 to 14 should be not be complementary, right. And this length of loop is about 8 nucleotides, and you will see a lot of variation in the microRNA depending upon the species or the evolutionary distance between the microRNA. Okay, as I mentioned, starts with A or U, right, and basically yes, so position 2 to 8 are part of the mature sequence, which is called the seed, and should be strongly conserved for the binding. So as we see also here, like this was the seed which should bind the mRNA sequence, messenger RNA sequence. Okay, so regarding how they are originated, so from the primary microRNA transcript, basically first of all Rocha acts on it and cuts, okay, this part. So you can see that the cutting is not even, it is uneven cutting, and forms the pre-miRNA. Okay, 5' and 3' are there, of which like this is more conserved and this is less conserved. Okay, and this is the loop which is about 8 nucleotides. Okay, and then basically the dicer cuts it and makes this miRNA complex like this. Okay, which is about 20 to 26 nucleotide long. These two loops are not perfectly aligned. Okay, there is an overhang of 2 base pair on both sides. Okay, which actually loads into the RISC complex and targets the mRNA, right. So this is how they originate. So to predict these, because they are regulating the gene expression or the mRNA basically. So so we have to predict these, and an estimation of these may help in understanding the regulation of mRNA in the genome. Okay, and they can also, like these days, serve as drug to target for various drug targets, and right. So and with the various disease pathogenesis, health, etc. So MIR base is one of the database that contains the MicroRNA sequences, right, and it's also contains a tool to predict the miRNA in the genome. Okay, so basically the database can be found at this site mirbase.org, which is the primary MicroRNA repository. Okay, and it contains information about all the hairpin portion of the mRNA transcript that we saw just now. Okay, and basically that is why it is called hairpin miRNA. Okay, the mature miRNA. So it does the homology based mapping against the miRNA. So basically blast have to be run, and it can be done at your local PC. So first of all, you have to download the database, right, and format it, and okay, after that basically you have to run the blast command to search against the database of mirbase, which is the hairpin, basically contain the hairpin information, hairpin MicroRNA information sequences. Okay, and then basically you will be doing against the blast okay, against your sequence, and specify the output, e value, like the way the blast is done, identity values, coverage, right, how many threads you want to use in your system, like how many cores you want to dedicate, right, for this purpose. And it gives you the output of the different type of MicroRNA, their percent identity, start, end, coverage, e value, and the score, like the way you get the blast result. This is the same blast result output. Okay, then MIR GeneDB is another MicroRNA gene database for animals, and can be used for prediction of MicroRNA. So these MicroRNA play a lot of roles in regulation basically, and in plants, animals, health. Okay, so you can say that in stress, in heavy metal, response to heavy metals, immune responses, nutrient deficiency, the different type of miRNA are produced, expressed, and they act on their targets and regulate the expression of those mRNA transcripts. Okay, similarly in case of cancer, these are very well known today, and people are trying to find various new or novel miRNA, which in case of cancer or other influenza and other kind of viruses also. Okay, so basically some could be activating or some could be inhibiting that particular mRNA. So these there are so many reports and publications these days that show the role of different type of miRNA, and also selected miRNA in various diseases, and and complex situation like cancer. Okay, so okay, again to give you an example of the same. So basically total tRNA was in the 2 genomes, for example 3 genomes, turmeric or banyan tree or people. So tRNA was total predicted was 679 and 668 in the 2 genomes, and in case of turmeric it was 1826. The microRNA was in the 195, 185, and it was 335 in case of turmeric, right? Total coding genes was 25,000, 26,000, 27,000, and like 50,000 in case of turmeric. So you can see, and the total ribosomal RNA was 1,135 and about okay, so ribosomal RNA not mentioned here. So basically, so you can see that there is not a fixed number that you will get. It will vary from genome to genome. The size may matter a bit, like more the or it may show correspondence with the number of genes, total number of protein coding genes. So coming to the end of this first part of this lecture. So in this part we covered the various tools that we can use to predict the various non-coding RNAs, okay, such as the tRNA, ribosomal RNA, miRNA, which are very important because a number of tRNA and miRNA and the ribosomal RNA are standard information that you always put in your manuscript. And to predict the microRNA in a genome can be really useful in understanding the type of microRNA related regulation that may happen in that genome, right. So overall, basically these numbers are reported in the manuscripts and in the analysis of any genome. And in the second part of this lecture, we will look at the functional annotation of genes using eggnog and KEGG pathways. And so we'll be covering those concepts in the second part of this lecture. Thank you.